Colorectal cancer.
In order to understand cancer, we must first understand DNA, how it replicates, and how proteins form. DNA is a double-stranded molecule which is made up of a combination of base pairs. They're adenine, thymine, guanine, and cytosine. The base pairs bind through hydrogen bonding and create the DNA helical structure. Adenine must bond to thymine, cytosine must bond to guanine, and they must bond in a specific order. The sequence of base pairs is read and transcribed into RNA and then translated into proteins. The parts of DNA that code for a protein are called genes. If the base pairs in a gene sequence are assembled or read wrong, a mutant protein will form that doesn't function properly. This is called a mutation. A mutation is defined as a permanent change in the genetic structure of DNA. Mutations can be passed from parent to offspring and are the leading cause of cancer. In fact, mutations are the only cause of cancer. Aside from genetic mutations, epigenetic mutations can occur as well. They are another kind of mutation defined as a change in gene expression without a change in gene sequence. This means that unlike genetic mutations that cause a changed gene product due to an error in base pairing, an epigenetic mutation is a change in the mature DNA with no change in base pairing sequences. The picture on the right shows common epigenetic mutations. The addition of certain organic molecules, such as the methyl group circled here, can mutate DNA and lead to a different protein. Epigenetic mutations are influenced by many different things. Factors during development, as well as environmental factors, drugs and pharmaceuticals, aging, and diet can influence epigenetics and potentially lead to mutations.
Genetic and epigenetic mutations are the basis for cancer and tumor growth because they change the way a cell functions and grows. In certain genes, a mutation leads to a change in cell growth rate. If the change causes faster, uncontrolled cell growth and division, then a tumor will form. With this in mind, it is important to note that not one single mutation can cause cancer alone. Multiple mutations are required. The mutations can be in one gene multiple times or in multiple genes. To illustrate this, I provided a picture to the right. A normal healthy gene is depicted to the left. The healthy gene creates a protein product that controls cell division and growth and ensures that tumors can't form. On the right, the same gene is depicted but with a mutation. The mutation is represented by the red bar. This mutation leads to a change in protein function that makes it unable to suppress and regulate cell growth. This can lead to cancer. The APC gene is the gene attributed to causing colorectal cancer when it mutates. APC is short for adenomatous polyposis coli and is an anti-oncogene. Anti-oncogene is also known as a tumor-suppressing gene. The protein product that a healthy APC gene creates inhibits cell growth. However, when mutated, the APC gene becomes inactive and no protein is able to form. When the protein is unable to form, unchecked and uncontrolled cell growth is allowed. The picture to the right shows how tumor-suppressing genes act as brakes, like on a car. When mutated, the brakes don't work properly and cells grow and divide rapidly, leading to cancer. The picture shows it only takes 2 mutations to lead to cancer, but that is just for the purpose of making the concept of mutations and cancer easier to understand. In APC genes, methyl groups are added to the promoter region of a gene. A methyl group is an organic group that consists of carbon and 3 hydrogens. The promoter region is a section of a gene that initiates transcription to create a protein from the DNA sequence. Methyl groups inactivate the DNA so that it can't make its corresponding protein product. In APC, this means that tumor-suppressing products aren't made and cells are allowed to grow rapidly, leading to cancers. The picture to the right shows histones wrapped up and either methylated or unmethylated. A histone is a group of proteins all wrapped up into chromatin DNA. Histones are tightly bound when they have methyl groups on them. This makes it hard for DNA to be transcribed into protein because the promoter region can't be reached. Highlighted here are the promoter regions of DNA. As you can see, in methylated DNA, they can't be reached and therefore transcription cannot occur. This picture sums up the preceding slides. The purple cells, shown here, are epithelial cells. Epithelial cells are found everywhere in your body. Since this depicts the colorectal cancer tumor growth, take these cells as being in the rectum or colon. The first step is a mutation in the APC gene. This causes methyl groups to bind to the promoter and inhibit gene function. Cell growth becomes uncontrolled, and more mutations occur. After many mutations, shown here, a cancerous tumor is formed